Good morning, everyone. Thank you, Francis, for the great introduction. I do not have to explain some of the philosophical underpinnings of using Omics. Let me just say that when we use Omics, let's not forget bioinformatics. And all of these tools require intensive training. And we're happy to say that in places like the Marine Science Institute and in the Philippine Genome Center, we already have such capabilities, and we can arrange workshops to train the rest of us who are classical microbiologists in these new methods. And we say that the methods we use are holistic. It's so important to be adept in the classical microbial methods, as we would like to demonstrate in this area of research that I am in, with a lot of young Filipino scientists, including the ones who succeed me in the Marine Science Institute, Lilibeth Salvador Reyes and Isadora Yu, our young faculty. And we shipworms, tamilok. How many know tamilok? It's edible. It's like an oyster, but not as cute as an oyster. Although one that we isolated recently or found in the Abatan River, it's a Lithoredo abatania. It's very cute. It's like an oyster. It's so white and translucent, except that it's a gut bacteria that take in methane. So it's very interesting. So I'd like to say that I'd like to recognize that it is funded by the US NIH, Fogarty International, and National Science Foundation. Only these types of funding agencies will support this kind of star-tracking research. And it involves US universities with the Marine Science Institute. And we've learned a lot from them, okay? And so I'd like to reiterate what Francis said: microbes are biological partners of most organisms, and symbiotic microbes can be integral to most processes. I would say we'd like to emphasize that microbes can be good, okay? They benefit us. So if I am in the field of drug discovery, I'd have to think of microbes that might produce compounds that may not be bad or so toxic to the host, a macro host like a human. And on the right, you see a figure from the publication by Blum on the human microbiome, and it's telling you what Francis said. In every part of your body, there's all these kinds of microbes. They have been classified. Okay, so there, the human body cartoon, and then at the bottom, you can't read it, but in the intestinal flora, there is already comparison of the healthy control and the patient for inflammatory bowel disease. We know that's important, related to, am I right, diverticulitis, and then to type 2 diabetes, and then to what they call this, necrotizing enterocolitis. So it's telling you that the microbes become different in these conditions, disease conditions. And I'm naturally interested in this because I'm in the drug discovery field. But on the left side, you'll see the sponges with their microbiomes. That will be the subject of Dr. Gisela P's talk later. And that's really where it all started in the marine environment, where sponges, which are the prolific producers of bioactive compounds, found to be full of microbes. Microbes coming in and out. Those drugs, those compounds, have the microbial origin. Thing is, some of the microbes are specialists. They have like one dominant bacterium, and others have many bacteria. And they're very important recent publications on these two types of sponges. But we're on shipworms today, and they're very important because they're mostly found in mangroves, and in fact they have an economic and an environmental function. So this is not just about drugs. It was our great hypothesis to think they could be useful source of drugs, but in reality what shipworms are known for is their major role in transferring energy from the terrestrial environment to the marine environment. back to the marine environment. Why? Because they have in their gills cellulitic bacteria. They have bacteria that produce a suite of celluloses or cazymes. And that's because, well, they break down wood. They live in wood, in mangrove wood. And so, well, we know that terrestrial has probably more, energy sources from the sun for photosynthesis. And we know that the environment is generally energy and nutrient limited. And so there is a way for these tamilocks and their bacteria to break down the cellulose, bring glucose back to the marine environment, and create niches or little habitats for marine organisms to thrive. So important. Another economic importance of tamilocks, it's devastation. Billions of dollars worth of wharves, of piers that are broken down, ships, wooden boats that are broken down with holes because of the tamilocks. Okay? But then we look at the good part of tamilocks. And so let's just take a look at all shipworms, gilled with abundant bacteria, shown with those circles. Generally, this is the anatomy of the shipworm, depending on where it specializes or how it specializes. Some have like a vestigial cecum. What's a cecum? It's like your GIT, the large intestine. Okay? And can you imagine in this wood-consuming shipworms, you have cecum, but in the kufus, which I will tell you about later, which is like this unusual, scary, elusive giant shipworm, it's vestigial. So it's reduced. Okay? And it's a cecum, large intestine, but it is quasi-sterile. Isn't that crazy? Large intestine, but quasi-sterile. No other bacteria except, well, a few. Okay, mangrove specialists, they have an anal canal, and there is secondary wastewater. It's a very efficient animal. And we look from the right to the left, you have the input of food in the mouth. You have the water coming in, and then waste coming out. Then you have the anal canal. So it's a very, very neat animal, and highly successful as well. So what are they? They are not worms. They are bivalves. They're like oysters and clams. And they have degenerate shells. So they're very unusual, and they adapted to their environment. On the left side is a giant, elusive shipworm, never before found. Centuries of a mysterious shipworm, now is cellulitic. And that's so important to us. But on the left side, we got a compound that we're now testing. We filed a patent application for it, Mindapirose, because it came from Mindanao, and it came from a bacterium from the kufus. Very important. It's only one of the compounds that we have been working on, okay? And this is work by my RA, who's now going for his PhD abroad. Okay, now, just to show you, we have to do the phylogenetics. So before you do genomics or metagenomics, make sure you know how to do phylogenetics. So you know how to culture bacteria, purify it, and then do DNA extraction and do your 16S. So there you have the new bacterium, which is a sulfur oxidizer. That is coded 2141P, and there is the phylogeny in the bottom of the Kufus polyphenolus. And there has to be a connect, some kind of connect. Okay, so what did we see in the paper? That in Kufus, there's really that major bacterium, the one with the high reeds, the red ones, okay? There's several strains of them, and they are interrelated, shown by their genomes. We had isolated each of these strains, and then we are able to show on the right, they are very similar, okay? And they are all thioautotrophic. They are sulfur oxidizing. Questions answered by Omics. What's the composition of bacterial symbionts? Who's there? As Francis said, what is the biosynthetic potential? That's my interest. BGCs, biosynthetic gene clusters, and all that stuff, okay? Because we want drugs from these organisms. So we are doing metagenomic mining, and so we have this paper. It's already out in the online, but it's actually not yet been accepted for publication, pending revisions. And I'd like to recognize the co-authors. And this is major work from 20 gilled metagenomes from 6 different host shipworm genera, including Kufus. including the wood-boring ones, and including the sediment-dwelling ones. Okay? And that's the general method summary, and this is the detailed methods. So there's got to be a lot of training done for us to be able to do this correctly. Okay? Otherwise, it's garbage in, garbage out. Shrimpworm gill metagenomes are characterized by a few dominant bacterial species. So I'll just explain this. Sulfur oxidizing isolates are the first two at the bottom, x-axis, and then the cellulitic isolates are the last four or three on the x-axis. And then you will now see their hosts at the top. And it's correlating with what you've learned about it in the environment. One is sediment dwelling, sulfur dioxide, sulfur oxidizing, and the other is cellulitic. So there is a correlation between genomics, metagenomics, and the functionality, okay, that you see in the environment. So this is like global approach analysis. That is what metagenomics will allow you to do. Compare the isolated genomes, meaning we cultured those bacteria, purified them, and got their genomes. And then also did the metagenomes from the host material. And so you're able to show that together, you know, this is a rich resource of compounds, because we are focused here on those genes that encode for the production of secondary metabolites. And you will see that there are unique ones, but there are also common ones. It's telling you that in the marine environment, there are many common strategies taken by all of these organisms that produce the same compounds, but there are also those that are specialists. So there is a really rich resource, you know, from this group. Widely shared GCSs are present across wood-boring shipworm genera and may be important in the wood-boring style. That's what we want to connect. We want to connect the omics data with the lifestyle, with the functionality. Okay? And so here we have things that pertain to secondary metabolism. Conclusions: shipworm microbiomes are rich sources of novel secondary metabolites. Widespread gene cluster families are potentially important to the shipworm. Wood-boring lifestyle are present and are currently priority targets for compound isolation and characterization. Example, think about it. So you have a dominant cellulitic bacterium there in your gill. Oh, what's going to happen if, you know, the cellulases transported to the cecum where the cellulose gets in, okay, from the wood? What if other bacteria, you know, will compete for the food, for the glucose? So there must be bacteria that can produce antimicrobials, right? So there must be this cellulitic bacterium that's also producing antimicrobial compounds, which we have shown. This is producing small molecules. So that's just our hypothesis. I think it's being proven right. Okay, so other new shipworms discovered. So we have one in Mabini. It's Tamilocus mabinia, so we named it that way in honor of Apollinario Mabini, found it in Mabini, Batangas, and it's Tamilocus, is now in the literature. And then we have this cute oyster-looking but rock-boring shipworm. Can you imagine the different kinds of lifestyles? We're so proud of ourselves that we are so unique. We have so much diversity in the humans. They have their own diversity. Okay? So, ongoing analysis, functional analysis of these new, very curious shipworms, and we also want to do viral analysis. Future work, metagenomes in the surrounding environment. We have a UPV master student of Michelle Malay who wants to look at the bacteria in Sultan Kudarat, the sulfidic environment. Culturable microbes in the surrounding environment. She's going to try to do that. Tough luck. Chemistry of that surrounding environment. But in light of the SNT foresight goals of the past, I think this is so important. Basic research is so important. It has impact on the environment. So we said that the shipworms are major players in the environment, in the mangrove setting, and also a major player now, I would think so, in the discovery of new drugs from the sea. So, I wanted to thank the relevant literature, published literature. Published literature from our group and other groups, but lots of literature from our group published. And I'd like to thank the U.S. government for the funding. Let me say that at this point, there is continuity in this work because Lilibet Salvador Reyes, Isadora Liu, they have funding from the UP system EIDR program, which we set up to continue this work beyond this program, because this is already ending, so we're just finishing up middle of this year. But we already have a new program led by the young ones, already funded by UP to continue the work. Thank you very much.